So depending on what kind of organism you are and what your biological sex is, some cells are going to return to interphase. Other ones are going to skip this interphase and go directly into the second process of meiosis. If you're an ovary haver like I am, your cells go back into interphase um, until it's time for ovulation, basically. The cells go back into interphase. They do not have an S phase in the interphase between meiosis 1 and meiosis 2. We are shutting down the DNA synthesis part of interphase. So I want you to think about this. I'm not going to tell you the answer. Why would replicating the DNA between meiosis 1 and meiosis 2 just defeat the purpose of meiosis, right? There's two purposes of meiosis. So if we replicated DNA, we would destroy one of those purposes. You get to tell me which one. So meiosis 2, whether we immediately go from meiosis 1 into meiosis 2, or we have this kind of temporary interphase with no S phase, meiosis 2 looks the same as mitosis. We're going to have prophase 2. We are going to take our spaghetti form DNA, pack it up into the chromosomes. We are going to get rid of the nuclear envelope. In metaphase 2, we are going to line up the sister chromatids, and those sister chromatids are going to be what separates in anaphase 2. And then in telophase 2, we're relaxing from Cheeto back to spaghetti, right? Going from chromosome to chromatin, reforming nuclear envelopes. And now at the end of meiosis, we have 4 daughter cells. Okay. All right, so let's talk about the results of meiosis. We have 4 daughter cells. These cells are going to be called gametes if we're talking about animals. If we're talking about plants or fungi, we call them spores. These 4 daughter cells are haploid. They have 1 set of chromosomes in each of those cells. Because of the crossing over and the independent assortment, each of these 4 daughter cells are genetically distinct from each other and from the original parent cell.